We use a variety of different methods for our initial analyses. As we get a little bit of information, we begin to prospect a bit, bioprospect, take the venoms apart, look into them in a little more detail, look for interesting compounds using some of the more sensitive techniques that are available fairly readily now. Once we have something that shows potential biochemical interest, then we begin to look at it biologically. Typically, this is using cells. Sometimes it's using tissue baths or whole animals. Occasionally, we use the crude venom. We are focusing primarily on purified toxins, but I'll talk about some preliminary work with the venoms themselves. If we look at venoms from different rattlesnakes, and this is just a whole bunch of different species, what we see is that they range from extremely toxic, i.e. it takes a very little amount to kill an animal, to those that are less toxic. And it turns out these have chemistry components or aspects of their chemistry that are rather different than the less toxic ones. We can also provide a molecular fingerprint of the venoms, that is, run them out on single-dimension gels, separate the proteins on the basis of size, and simply ask what's in there, how complex in it is this secretion. So where we have these little letters at the top, those are individual samples of venoms. They're run out here. The big things are up here. The little things are down here. And what you can see just looking across that, there's a lot of stuff in there. Lots of things going on. We know enough about many of these compounds now that we can overlay this with labels. And so we've identified most of the components that are found here. And as you look at these various patterns, we see that they're differential patterns. Some snakes produce these compounds, some produce others. Well, that tells us there's a lot of variation out there, and in fact that variation is something that's very, very useful, because it turns out that some of the compounds that are down in this range that are absent here and are present here, those vary from species to species, and some of those have chemical properties that we may be able to capitalize on. We use a variety of different types of methodologies to pull apart these venoms. This is a little snake from down south of Lyman. We've done a lot of work on the eastern plains of Colorado. One of my other jobs, if you will, is to do herpetological surveys in conjunction with Colorado Division of Wildlife. And so we've had people out in the field all over the eastern plains since about 1995 looking for snakes and other small animals. In addition to finding those snakes, then we also collect their venoms when we bring them back temporarily. MALDI-TOF, this is a mass spectrometry technology that allows us to take a look at compounds in very high degree of precision, take things apart. We can use a variety of involved techniques for going in and taking some of the complexity we see in the venom in the single gel and pulling it apart, getting some identification of some of these compounds. And this is a snake that one of the undergraduates working with me presented her results at a meeting, national meeting in St. Louis last year. It's a snake from Mexico. We've collaborated with a group in Mexico City on that one. The Massasauga rattlesnake, this one that's found down near Lyman, has been in fact a very important and interesting animal for a number of different studies that we've done in conjunction with people in Singapore. So a snake from Colorado has made it, at least its parts have made it as far across the world as Singapore. And it turns out that there are a wide variety of these very specific little toxins in its venom. We've done some analysis of how these relate to other snakes, because it turns out that most of those type of toxins are found in the really dangerous snakes, the ones that are found in Australia, the ones Steve Irwin used to play with, cobras and other things like that. And so it's a very interesting group of toxins, and we're some of the first to identify these in local rattlesnakes. We've also worked with a snake that is problematic in one of our territories, that is the brown tree snake, an invasive species that has led to the demise of birds and lizards and small mammals on the small island of Guam in the Marianas. This molecule is very unusual. It's found as a very large amount of the venom component. Turns out that it's extremely toxic to birds and to lizards, but to mammals it's essentially non-toxic. Hundreds of times the amount of toxin, and there's no effect at all. If you're a pharmacologist or a biochemist, that becomes really fascinating. How does it knock out these type of animals, which are vertebrate animals, but it doesn't affect these. And that suggests then that we've got something very interesting here for probing neurotransmission, for probing how nerves and muscles communicate. That leads to the question, why work with toxins? Toxins are bad, right? Well, it turns out that poisons and drugs just differ only in how much you actually consume. And something that we would call a poison could be a useful drug at a very low dose. And so dosage is extremely important, as well as where this occurs and when. Most of the components in snake venoms are derived from normal regulatory components of our own bodies and the bodies of other animals. It's just when a snake gives it to you in a bite, it gives you perhaps 100,000 times as much as would normally be found in your blood vessels or in your body at that particular point in time. And that's not so good. Specifically, these toxins, though, can help us understand how drugs work, how compounds work, and in fact, they can lead us to designing drugs that are based on these big molecules. Proteins are pretty good sized molecules. Most of the drugs that we take are very, very small. And so if we can look at that big protein that's been designed by nature and say, there's a little piece right here that's really where the action's happening, then we can model a small drug that will work very effectively and quickly based on how these toxins interact with a particular target. And the way that they target things in the body is frequently extremely specific. What is the benefit to this? Well, higher potency of our drugs, less side effects, so a lot better efficacy, if you will, of drugs in general. And so there's a lot of potential here for looking at these venoms as basically a way to ask questions about what kind of drugs could we design. We've been doing some work with this little snake here. It's called a Puerto Rican Racer. It's one of these rear fang snakes that a colleague sent to us. He's working on one of these Caribbean islands. Unfortunately, we haven't had the chance to go down there. I really would think that we need to get out in the field just because it might be a good thing. Anyhow, we've again done this same kind of separation of the venom, get at this molecular fingerprint. We can actually cut out bands of interest here, chop them up with specific proteases, send them in for analysis, and get identification of what they are. And this one in particular, what we call AP11, is a type of molecule that interferes with how cells attach to one another and communicate with one another. And that turns out to have some very important implications for cancer metastasis, how cancer cells move around. Okay? We also identified that we had something, a blood contaminant up here. And so, again, this provides a turnaround analysis that's very, very rapid.